Hello students, welcome back to my channel. Today I am starting a new video and this is the third lesson, that is third part of the topic absorption by roots. So without wasting time, let's get started with the video. So today I will be talking about the tonicity. So there are basically three types of solutions: isotonic, hypotonic and hypertonic solutions. Now what do you mean by isotonic solution? Isotonic solution means iso means same. Iso means same. That means in this type of solution, the concentration of solute and the concentration of solvent both are similar. That means on both sides of the solutions, they have equal solute concentration. That type of solution is known as isotonic solution. The relative concentration of the water molecules and the solute on either side of the cell membrane is absolutely same. So in such condition, what will happen? There will be no net movement of water molecule. So no osmosis will occur, because on both the sides of the cell membrane, the concentration is absolutely same. The concentration of solute and the concentration of solvent both is same. So that's why no net movement of water will occur. So no osmosis will occur. This type of solution is known as isotonic solution. Now what will happen in case of hypotonic solution? Hypo means lower. Hypo means lower. That means in this condition, the solution outside the cell has a lower solute concentration. That means there the solvent concentration, the concentration of water is more than the fluids which is present inside the cell. So here the solute concentration is lower. That means the solution in which the cell has been kept, there the solvent concentration or the water concentration is more than the fluids which is present inside the cell. So as a result, what will happen? We know that osmosis means from their higher concentration to their lower concentration. So water molecules will move inside the cell. From outside it will move inside the cell. So basically endosmosis will occur. So here when a cell will be kept in hypotonic solution, endosmosis will occur. So again I am repeating, hypo means lower. That means in that type of solution, what will happen? Solute concentration is lower. That means solvent concentration or water concentration is more than the fluids that are present inside the cell. So as a result, what will happen? Net movement of the water will occur. Where to where? From outside of the cell to inside of the cell. So basically endosmosis will occur. Next is the hypertonic solution. Hyper means more. That means in this type of solution, the solute concentration is higher. That means in this type of solution, the solvent concentration or the water concentration is less than the fluids which are present in the cell. Again, we know that osmosis means from higher concentration to lower concentration. So where the water concentration is more? Inside the cell. So now in this case, what will happen? There will be a net movement of water. From where to where? From inside the cell to the outside. That means the water will move outside the cell. So exosmosis occur. So whenever a cell will be kept inside the hypertonic solution, exosmosis will occur. Why? Because hyper means higher solute concentration will be there. That means the solvent concentration or water concentration of that solution is lesser than the fluids which are present inside the cell. So as a result, water concentration is more inside the cell. So water will come outside from the cell. So exosmosis will occur. Got it? I think it's clear. So this is basically the concept. Whenever a cell will be kept inside an isotonic solution, then there will be no change in the shape and size of the cell. Whenever the cell is kept inside the hypertonic solution, then what will happen? Hypertonic solution, whenever it will be kept, all the water content of the cell will come outside. The cell will shrink in size and it will lose its shape. And whenever the cell will kept inside the hypertonic solution, then what will happen? The cell will slightly enlarge or even it can burst. Okay, see, this is the example that I am showing. I have shown an example of RBC, red blood cell. It is also a cell. So see what happens. In isotonic solution, what happens? Isotonic solution means the water concentration is absolutely the same outside and inside the cell. So there will be no net movement. So there will be no change in the shape and size of the cell. But what happens in hypotonic solution? Hypotonic solution means the water content of the solution is more than that of the content which is present inside the cell. So more and more water will enter. More and more water will enter inside the RBC or cell. So at one point of time, what will happen? The cell will slightly start enlarging, and one point of time it can even burst because it will not be able to hold any more water inside it. This is known as the turgid condition. Later on in this video, I will be talking about the turgidity. So whenever a cell will be kept on the hypotonic solution, then more and more water will move inside the cell. So at one point of time, it can even burst because it will not be able to hold any further water. And when the cell is placed in hypotonic solution, what will happen? The water content inside the cell is more than the outside. So more and more water will move outside. As a result, the cell content will shrink. There is a shrinkage of protoplasm. The cell shape will shrink. It will lose its shape and the size of the cell will shrink. So this happens. So this is the three conditions that I have shown with an example of RBC. I think now this total tonicity portion, this concept is absolutely clear. Next, we will move on to the turgidity and flaccidity. So what is turgidity? Basically, turgidity is the condition of the cell when it is fully extended. That means more and more water is going inside the cell. Just I have shown in this condition. This is a turgid cell. More and more water is entering inside the cell. Okay? Then we will call it as the cell is turgid. This condition will occur when the cell will be kept in what? In hypotonic solution. So basically due to the endosmosis of the water, the cell will enlarge and at one point of time it can even burst. So the condition of the cell when it is fully extended, when the cell will not be able to hold any further water, that is known as the turgidity. The cell wall is absolutely rigid and stretched by an increase in the volume of the vacuoles. Why? Due to the absorption of water. So whenever the cell will be kept in hypotonic solution, this turgidity will occur. Then we will call it as turgid cell. So it is basically occurring due to endosmosis of water. And in case of flaccidity, the cell content will shrink. There will be shrinkage. Why? Because exosmosis of the water, when it is kept in hypertonic solution, more and more water is coming out from the cell. So there will be exosmosis and there will be a shrinkage of the cell. Then we will call it as flaccidity, or even we can say it as plasmolysed cell. I am coming to this in the later on this video. Okay. So I am showing an example of turgid and flaccid cell. See, this condition is turgid cell. A well-potted plant, fully extended leaves are there because the vacuoles are full of water. But see the drooping down of the leaves in this plant, in this case. Why? Because all the water from the plants has come out. So there is no rigidity in the cell wall because exosmosis has occurred. All this water content has been given out from the plant or from the cell. So there is no more water inside the plant. So that's why the leaves have been drooped, or wilting of the leaves, you can say. So this is the example of a turgid condition. This is the example of a flaccid condition. I think the turgidity and flaccidity is absolutely clear. Now come to the turgid pressure and the wall pressure. It is very easy. Turgid pressure is the pressure that is exerted by the cell content on the cell wall. This you need to remember. And wall pressure means the pressure which is exerted by the cell wall on its contents. This is the basic thing. So turgid pressure will occur due to the turgidity of the cell. When the cell will become turgid, when the cell content will give a pressure on the cell wall, then that is turgid pressure. And when will wall pressure will occur? When the cell wall will give a pressure. Suppose this is the cell wall. So when cell wall starts giving the pressure on the cell content, that is known as wall pressure. Actually, this wall pressure maintains the structure of the cell. It keeps the cell in rigid condition. So turgid pressure and this wall pressure, this is basically the vice versa, the reverse thing. Target pressure means the pressure which is exerted by the cell content on the cell wall, and wall pressure means the pressure which is exerted by the cell wall on the cell content. Got it? It's very simple. Now we are coming to the very important part, that is plasmolysis. Most of the question from this chapter comes from this plasmolysis part. So what will happen? This is a cell. This is a cell. This is a target cell full of water. Now this has been kept in hypertonic solution. In hypertonic solution, where the solute concentration, I can say the water concentration of that solution is less than the cell. The cell contains more water than that solution. So if we keep this target cell in hypertonic solution, then what will happen? Slowly and slowly, see, the water will come outside. The water will come outside from the cell, and ultimately there is a shrinkage of protoplasm. There is shrinkage of protoplasm. This is very important. This term you should mention. There will be shrinkage of protoplasm, and this condition is known as plasmolysis, plasmolysed cell. Whenever there is shrinkage of protoplasm, all the cell content, the fluid, the water has come out from the cell. When the cell is kept in hypertonic solution, then that is known as plasmolysed cell, and the condition is known as plasmolysis. Now what will happen if we keep this cell, if we keep this particular cell, the plasmolysed cell, in hypotonic solution, then what will happen? Again it will move back to this condition, this particular condition. It will become normal. That is known as deplasmolysis. Deplasmolysis. That means we are neutralizing the plasmolysed condition of the cell. That is why the condition is, or the method is known as deplasmolysis. So the question comes, how we can recover this condition? Through deplasmolysis. If we keep this cell inside a hypotonic solution where the water content is more than the cell, then the water will move inside and this condition will be reached. This is known as deplasmolysis. I think the plasmolysis and deplasmolysis is absolutely clear. So again I am repeating, whenever a cell, like a target cell, is kept inside the hypertonic solution where the water content is less than the cell content, then what will happen? More and more water will come outside from the cell. As a result, there will be shrinkage of protoplasm due to exosmosis. So this condition will be reached, which is known as plasmolysed cell. This process is plasmolysis. Now how we can get back to this normal condition? If we keep this cell inside a hypotonic solution where the water content is more, solvent content or concentration is more than the cell, then endosmosis will occur. More and more water will move inside the cell, and then we can get back to this target condition, target cell. This is known as deplasmolysis. Got it? Okay. Now see, often a question comes that salting of meat or addition of salt is very much required in pickles. Why? The answer is due to plasmolysis. Why? Because when we add salt, then the bacteria which are present inside the meat or the pickles, they are killed because water is drawn out from the bacterial cells. Due to exosmosis, there is shrinkage of protoplasm inside the bacterial cell. So they will die, and we can get pickles fresh for a longer period of time, and meat also. So this is an application of plasmolysis in daily life. So in ICC question can come give reason why salting of meat is required, why addition of salt to pickles is very much required. So this type of practical question can come in ICC. It often comes rather. Okay. Next is the root pressure. Now see, root pressure, basically it is the pressure which is developed in the root due to the continuous inward movement of the water. This is the root hairs, and this root hair actually, they continuously absorb water and minerals from the soil. So due to this continuous inward movement, a pressure starts creating inside the root due to the cell to cell osmosis. From one cell to another cell, the water is continuously carried on. This is the ascent of sap, we can say. So this root pressure, due to this root pressure, the water is actually moving up from the roots to the cell, to the leaves. This root pressure is responsible for this. See, root pressure is the pressure which is developed in the roots due to the continuous inward movement of the water through cell to cell osmosis, which actually helps in the ascent of cell sap upward through the stem. And just because due to this root pressure only, the cell sap actually rises in the tube or through the stem. Now if there is a cut or any injury in the stem also, we can see there is a loss of water, that is cell sap. This is known as bleeding. Often question comes in them the following. How we can find that there is a root pressure? If there is a minimum or minute cut or injury in the stem, then we can see that a watery liquid, watery substance, that is cell sap, is coming out through that injured part of the stem. That is known as bleeding. So there is a continuous pressure. That is why if there is an injury, the water is coming out. This root pressure is actually responsible for the carrying of the water through the roots to the stem. Okay, this root pressure is responsible for the upward movement of the water. So that's it. I think all the portions that I have covered today in this video is absolutely clear to all. Still, if you find any problem or if you have any doubt in any portion, then please do comment me in the comment section. I will try to clear all your doubts. And only another part is left, where in the next video I will discuss about some important experiments from this chapter, and then this chapter will be done. So hope you have liked this video. If you think that this video is helpful and informative, then please like my videos and do share my videos with your friends. And if you haven't yet subscribed to my channel, then please subscribe to my channel and don't forget to click the bell icon, because if you don't hit the bell icon, then you will not get notifications immediately after I upload a new video. So I will be back with a new video very soon. Till then, stay tuned to my channel, and thank you for watching the video.